Hello everyone. In the last module, we saw how the mature seed is formed. Seeds represent a stage in the plant's life that decides the fate of the next generation. They enable the plant to overcome harsh environments and give rise to an offspring when conditions become favorable. Plants have evolved a variety of seed dispersal methods to ensure reproductive success. Seed dispersal may take place by wind, water, insects, or animals, including human beings. Since plants cannot move, the ability of seeds to be dispersed as far away as possible from the mother reduces the competition for resources and also enables the species to migrate and colonize new geographic regions. In this module, we shall see how the dispersed seed starts the next generation through the process of germination. The learning objectives of this module are understanding the various types of dormancy shown by seeds, how dormancy is overcome to initiate germination, and finally, how the processes of dormancy and germination are regulated. Let us start now. The seed at the time of dispersal shows very low moisture content, minimal metabolic activity, and an embryo that is maintained in a quiescent or dormant state. Seed dormancy can be considered as a temporary failure of a viable seed to germinate under conditions that are otherwise favorable for germination. It may be due to the underdeveloped state of the embryo at the time of seed dispersal. A dormant period is required for the embryo to complete its development before it can germinate. This is called morphological dormancy and is seen in many palms which need about a month after dispersal to complete embryo development, only after which they can germinate. Seeds of many members of Fabaceae or legumes have a hard seed coat and can germinate only when the seed coat of the mature seed is removed or punctured. This is called physical dormancy and is imposed by the maternal tissues like testa or endosperm tissues surrounding the embryo. Seed coat thickness and the deposition of polymers such as tannin and suberin in it enable the seed to remain in a dormant state. Now, seeds of a majority of species exhibit a type of dormancy called as physiological dormancy. This arises due to the accumulation of the plant hormone abscisic acid, which prevents germination until specific environmental cues have been perceived by the seed. Any factor that provides the environmental requirements for germination brings about a release from the dormant state. Seeds can be grouped into two types depending on their hydration status. Non-dormant or recalcitrant seeds have about 60% or more water content at the time of dispersal and lose viability if the water content drops below this level. Dormant or orthodox seed show less than 10% water content and are desiccation tolerant. Now, germination proceeds through three phases. First is rapid water imbibition. Second is reactivation of metabolism. And third is the protrusion of the radicle and its growth. Seeds at the second phase can be dried and stored again. That means seeds that have been imbibed can again be dried and stored. But at the third phase, when radical protrusion occurs, the germination process is irreversible. So, radical emergence represents an important developmental switch for seeds, which subsequently leads to seedling establishment. Now, recalcitrant seeds can successfully complete the transition from phase 2 to 3 of germination when conditions are favorable, but can switch off the metabolic processes under unfavorable conditions. Because of their hydration status, recalcitrant seeds cannot remain viable for a long period. Orthodox seed however, which show very low water content, are metabolically inactive. These seeds can be stored for a long period, especially under cold temperature, without losing their viability. Whether a seed will remain dormant or proceed to germinate depends on the environmental conditions. Recalcitrant seeds germinate immediately when environmental conditions are favorable. However, under unfavorable conditions, recalcitrant seeds gain dormancy as a protective measure. This is called secondary dormancy and can be lost and gained repeatedly until either germination occurs or the seeds lose viability. The potential for dormancy has to be overcome in orthodox seed when the environmental conditions are favorable for germination. Such seeds show a transition from the dormant state to a non-dormant state, but do not germinate till the passage of an appropriate after ripening time. After ripening, therefore, is a time and environment regulated process occurring in the dry seed. Germination is not triggered immediately in the seeds but occurs after the passage of an appropriate after ripening time. In several cereal species, this after ripening period is important for preventing the seeds from germinating while they are still in the panicle, especially under moist conditions which would otherwise favor germination. Hence, after ripening determines whether the seeds progress toward germination or remain dormant. The process of germination represents a sequence of events that occur before radical emergence in imbibed non-dormant seeds. After imbibition of the seed, the hormone gibberellic acid initiates the breakdown and mobilization of carbon reserves from the endosperm or cotyledons by inducing hydrolytic enzymes. The hydrolytic enzymes may be amylases, which convert starch to glucose, or proteases, which convert proteins to amino acids, or lipases, which convert the triacylglycerols to the fatty acids. These events are followed by the synthesis of cell wall loosening enzymes and proteins like expansins and subtilicin, which facilitate radical emergence by bringing about seed coat rupture. Abscisic acid and gibberellic acid levels are important for keeping the seed in a dormant state or in bringing about a release from dormancy to facilitate germination. We shall see something more about the role of these two hormones in either maintaining dormancy or facilitating germination. We have already seen in the earlier module how ABA is synthesized and how ABA signaling occurs to bring about the synthesis of storage reserves and to protect the seed against desiccation stress when it loses water. ABA also plays an important role in the expression of seed dormancy. We have seen that the enzyme NCED or 9-cis-epoxy carotenoid dioxygenase, which forms xanthoxin, is a major regulatory enzyme in ABA synthesis. While the cytochrome P450 monooxygenase CYP707A brings about the catabolism of ABA by hydroxylating it. After imbibition, a decrease in ABA levels is observed mainly due to CYP707A activity which promotes germination. ABA perception and signaling also play an important role in the induction and breaking of dormancy. We have studied the ABA signaling pathway in the last module. Just to refresh your memory, ABA signaling through binding of ABA to the PIR-PIL-RCAR receptor inhibits the protein phosphatase PP2C and leads to phosphorylation of the SNRKs. The phosphorylated SNRKs in turn phosphorylate the ABA-dependent TFs or transcription factors. These TFs bind to abscisic acid response elements or ABREs present in the promoter sequences of ABA-inducible genes. One such ABA responsive TF is the ABA insensitive 5, which inhibits germination under unfavorable environmental conditions. It does so by repressing the expression of phosphate 1 or the PHO1 gene, which is involved in phosphate transfer from cotyledons to radicals and leads to germination. The second gene regulated by ABI5 is the gene coding for the polygalacturonase inhibiting proteins or the PGIP. PGIPs inhibit polygalacturonase activity. Polygalacturonase brings about the degradation of pectin in the seed coat or testa and leads to seed coat rupture to promote germination. ABI5 induces the expression of the PGIP coding genes, thereby inhibiting germination. While ABA levels decrease during imbibition of seeds, an increase in the levels of gibberellin or GA is seen at this time. This hormone plays an important role in promoting germination. Biosynthesis of gibberellic acid involves three cellular compartments, namely the plastids, endoplasmic reticulum, and the cytoplasm. In the plastids, terpene synthesis join 4 isoprene units to form the 20-carbon intermediate geranyl-geranyl diphosphate. This is converted to corine and exported from the plastids to the ER, where it is oxidized to form GA12. GA12 is then acted upon by two oxidases, GA13 oxidase and GA20 oxidase, to form the different GA types which show variation in the side groups attached to the tetracycline gibbene skeleton. The bioactive GAs are formed by the action of the GA3 oxidase enzyme, while the GA2 oxidases inactivate the bioactive GAs. The activities of GA3 oxidase and GA2 oxidases are important in regulating the levels of bioactive GA during the process of germination. GA perception and signaling pathway regulates the expression of GA-inducible genes, which play an important role in germination. The GA receptor, called GA-insensitive dwarf or GID1, is a soluble protein localized to both the cytoplasm and the nucleus. The GA-GID complex has the ability to interact with proteins called DELA proteins. Now, the DELA proteins are nuclear transcriptional regulators that function as negative regulators by inhibiting GA signaling. DELAs do not have a DNA binding domain, and they exert their growth repressive effects by interacting with transcription factors that are positive regulators of GA responses. So, as seen in the figure, DELAs attach to the transcription factors, and when DELA is bound to the transcription factor, no transcription takes place of the GA-inducible genes. However, when the GID-GA complex binds to DELA, then the transcription factors are left free to bring about the transcriptional regulation of GA responsive genes. The DELA, which is removed from these transcription factors, is then degraded through the 26S proteasome pathway. The transcription factors bring about the expression of genes related to cell wall modification like expansins and pectin esterases. They also activate the genes involved in the expression of a variety of hydrolytic enzymes to remobilize the reserves during germination. To bring about the rapid activation of processes related to germination, the dry desiccated seeds also have a large number of long-lived mRNA species which can be translated immediately after imbibition. The germination process is completely halted by application of cycloheximide, which is a protein synthesis inhibitor, but not with the transcription inhibitor actinomycin B. This indicates that the stored mRNAs have an important role during the early processes of germination before transcription activity starts. Many stored mRNAs code for ribosomal proteins, translational initiation and elongation factors of protein synthesis, etc. The amount of long-lived mRNAs then reduces after the start of germination and the synthesis of both proteins and RNA is eventually initiated. The mRNAs coding for the ABA-related and late embryogenesis abundant or LEA proteins, which are expressed during seed maturation, are some of the long-lived mRNAs stored in dry seeds. These are thought to play a role in the expression of secondary dormancy, which is observed in recalcitrant seeds. We have already seen that recalcitrant seeds can undergo several cycles of dormancy and release of dormancy, which is called as secondary dormancy. And so these proteins play a role during the process of induction of secondary dormancy. The mRNA for gibberellin receptor GID1 is also a long-lived mRNA, probably because early GA signaling is important for the initiation of germination. An interplay between abscisic acid and gibberellic acid is therefore a key regulatory factor in regulating dormancy and germination. ABA synthesis and signaling, along with GA catabolism, dominate the dormant state, while GA synthesis and signaling, along with ABA catabolism, dominate the transition to germination. The complex interplay among hormone synthesis, degradation, and sensitivities occurs in response to environmental conditions and can result in either dormancy cycling or germination. Ambient environmental factors like light, temperature, and nitrate affect the ABA-GA balance and the sensitivity to these hormones. Let us see something more about how these environmental factors affect the abscisic acid and gibberellic acid levels during germination. Abscisic acid and gibberellic acid interact with various environmental signals like light and temperature in coordinating the germination process. Light becomes vital after germination because the seedling has to carry out photosynthesis to survive once the seed reserves are spent. Light perception by the photoreceptors called phytochromes, which are sensitive to red and far red wavelengths of light, plays an important role in seed germination. Phytochrome A and phytochrome B are chromoproteins which occur as dimers. The chromophore is made up of the byline pigment. Phytochromes exist in an inactive PR form in the dark or in far red light of wavelength around 720 nanometers. This PR form is converted by red light of wavelength around 660 nanometers into the physiologically active PFR form. PFR can again be reverted into the inactive PR form by absorption of far red light. This photoreversibility between PR and PFR form is seen in phytochrome B, but the PR form of phytochrome A is degraded and cannot revert back to the PR form. Phytochromes are known to modulate the timing of seed germination. Absence of light may indicate burial under the soil, And many plants also use the red far red ratio to detect the presence of shade of a leafy canopy and prevent germination. Phytochrome A senses very low fluence light which may reach the seed lying under the soil and triggers the photoinduction of seed germination. In contrast, phytochrome B mediates a photoreversible response to red and far red wavelengths at higher intensity light and also shows photoreversible effects on germination. Phytochrome proteins, which are present in the cytosol in the dark, translocate to the nucleus in the PFR form under light. In the nucleus, the PFR form regulates the transcription of a number of genes that mediate plant light responses. Phytochrome-mediated light regulation of seed germination occurs partly by binding of the phytochrome protein to the transcription factor phytochrome interacting factor or PIF1 and inactivating it by phosphorylation. The phosphorylated PIFs are then degraded through the proteasome pathway. PIF1 is a repressor of several genes involved in ABA and GA biosynthesis and signaling. Phytochromes increase bioactive GA levels in germinating seeds by the light-induced activation of the transcription of GA3 oxidases, which convert GA precursors to bioactive form, and repression of the transcription of GA2 oxidases, which catabolize bioactive GAs to the inactive forms. Phytochrome mediates also the expression of GA signaling DELA proteins through PIF1. So the GA signaling DELA proteins like GAI or RGA, which prevent the expression of GA responses, are not allowed to express due to degradation of PIF1 by the PR form of phytochrome. Phytochrome-mediated regulation of ABA metabolism and signaling is seen to occur by the downregulation of transcription of the ABA biosynthetic gene NCED and upregulation of the ABA catabolism genes ABA8 hydroxylases and CYP707A. The freshly harvested grains of many cereals are not responsive to red far red light, but show a strong inhibition of germination under blue light, though they do germinate in the dark. The blue light-induced dormancy is completely lost in after-ripened grains, which germinate equally well in light or in darkness. After ripening is important to prevent seeds from germinating while they are in the ear itself under moist conditions. Blue light is seen to inhibit germination in these freshly harvested grains through induction of NCED expression and inhibition of ABA hydroxylase activity. After ripening nullifies the blue light effects by activating ABA catabolism and GA biosynthesis genes in imbibing grains. The blue light response is mediated through the flavoprotein photoreceptor cryptochrome 1. Cryptochrome 1 undergoes blue light dependent phosphorylation, which is important for its activity. Since the timing of seed germination is crucial for optimizing growth of the seedling, plants seem to use multiple systems for sensing the light environment over a wide spectral range in order to enhance plant survival in a fluctuating environment. In temperate regions, dormancy is relieved by exposure of seeds to a period of chilling temperature in the dry dormant state. Seeds experiencing moist chilling in the soil during winter enter secondary dormancy and delay germination until the following spring. Similarly, cold treatment given to dry seeds, which is called cold stratification, is known to promote germination upon imbibition by breaking down dormancy. In the seed coat as well as in the embryo, abscisic acid levels decline during moist chilling, and this has been shown to increase the germination capacity of seeds when eventually exposed to ambient temperature. In seeds that are kept at ambient temperature after subjecting them to moist chilling, an inhibition of ABA biosynthesis enzyme NCED and the modulation of ABA signaling genes is observed. Another hormone, jasmonic acid or JA, is also induced during imbibition following cold exposure. JA is a cold stress response hormone. It inhibits the ABA biosynthetic enzyme NCED. Dormant imbibed seeds of temperate plants not subjected to this cold exposure maintain high ABA levels and do not germinate efficiently at higher ambient temperature. It has been seen that cold stratification can be substituted by application of GA to dormant seeds, suggesting crosstalk between the cold and GA pathways. It has been observed that GA3 oxidase, an important enzyme in the biosynthesis of active GA, is induced by a cold stratification treatment. In addition to pure seasonal cues like light and temperature, the levels of nitrate in the soil also plays an important role in regulating germination. Nitrate levels in the soil are higher in the absence of a canopy, and the soil nitrate works as a key signal molecule by interacting with hormone metabolism in seeds. High nitrate application to plants also brings about an increase in the endogenous nitrate content of seeds, which leads to dormancy release and lower abscisic acid levels in the seeds. In Arabidopsis, the nodule inception or NIN and the NIN-like protein or NLPs are a family of transcription factors that are involved in nitrate signaling. NLP8 is a key nitrate sensor and promotes germination in the presence of nitrate by direct transcriptional regulation of CYP707 and a corresponding decrease in abscisic acid levels in response to endogenous nitrate. Besides nitrate, ABA signaling is also attenuated by reactive oxygen species and nitric oxide, whose levels increase when the seed coat ruptures and the oxygen content of the seed rises. Hence, the reactive oxygen species and nitric oxide counteract the positive effect of ABA on seed dormancy maintenance. Hydrogen peroxide, which is a reactive oxygen species, and nitric oxide have been shown to accumulate rapidly upon imbibition and precede the induction of genes regulating ABA catabolism or GA biosynthesis. We have seen how germination is promoted by various extrinsic factors like light, cold, and nitrate, and intrinsic factors like abscisic acid and gibberellic acid. All these factors bring about radical emergence from the seed. Post-germination development involves the opening, greening, and expansion of cotyledons, marking the switch to autotrophic development. Seedlings growing in darkness display scotomorphogenesis or etiolation, which is characterized by elongated hypocotyls, apical hooks, pale cotyledons, and short roots. Scotomorphogenesis enables the seedling to rapidly grow within the soil in darkness. In the dark, several signaling pathways activate growth but also suppress the activity of proteins that are involved in photomorphogenesis. The constitutive photomorphogenesis or the COP1 protein, which is part of a larger E3 ubiquitin ligase complex, plays an important role in the suppression of photomorphogenesis in the dark. This ubiquitin ligase complex is a part of the ubiquitination pathway which marks proteins that are to be degraded through the 26S proteasome pathway. COP1 mutants show a phenotype of light-grown seedlings, that is, they show a loss of apical hook, open cotyledons, and shortened hypocotyls even in the dark. In dark, photoreceptors are inactive and COP1 is largely present in the nucleus as part of the E3 ligase complex, which target photomorphogenesis promoting transcription factors such as elongated hypocotyl 5 or HY5 for ubiquitination and degradation through the 26S proteasome pathway. The plant hormone gibberellic acid plays an important role in cell expansion during hypocotyl growth, both in the light and in dark. It has two different functions in seedling development in the dark. Firstly, it promotes the scotomorphogenic processes such as hypocotyl elongation, and secondly, it represses the expression of photomorphogenesis in the dark by negative regulation of light-induced genes. Like COP1, mutants impaired in GA synthesis show the de-ethylated phenotype in the dark. In the dark, GA concentration is usually higher, and GA binding to its receptor GID1 brings about ubiquitination and proteasome degradation of DELA proteins. In the absence of DELA proteins, some of the PIF proteins, which are positive regulators of genes involved in elongation of cells, lead to rapid growth of the hypocotyl. Hence, GA seems to regulate both the COP1 and the PIF1 pathway during scotomorphogenesis. On exposure to light, COP1 remains in the cytoplasm, and HY5 can therefore activate the expression of photomorphogenesis related genes. In addition, the PFR form of phytochrome leads to the degradation of PIF proteins and brings about photomorphogenesis. At the end of germination, a plant becomes established as a seedling, which shows an increase in size and complexity as it develops to give rise to a plant with roots, stem, and leaves. We shall study the development of the vegetative plant body in the next module.